Hi, it's Dr. Phoenix. Today we're going to be talking about protein synthesis. Protein synthesis is an extremely important topic because we are basically built from proteins. All of our body structures and all of our traits are a result of proteins. And all the chemical reactions that take place within our bodies and within our cells are a result of enzymes that drive these chemical reactions. And of course, enzymes are proteins as well. So this topic, protein synthesis, is critical to our understanding of biology. So protein synthesis begins at the gene level. Remember that genes are part of DNA, and they are the part of DNA or a part of DNA that codes for one particular protein. So there are many genes, hundreds to thousands of genes on each DNA molecule within our cells. Each gene codes for one particular protein. Now, in order to carry out the instructions of these genes, there are RNA molecules that have to do the job. DNA, and therefore the genes, is in the nucleus of the cell. And protein synthesis takes place out in the cytoplasm where the ribosomes are. So since DNA can't leave the nucleus, the message that DNA is carrying, those blueprints, have to be carried out into the cytoplasm where protein synthesis takes place. So there are three main players in this process, and they are all molecules of RNA. These three RNA molecules are referred to as messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA is the molecule that actually copies the code or the message or the blueprint from DNA and takes that message out into the cytoplasm. Ribosomal RNA mixes with proteins and actually makes the ribosomes where the protein synthesis takes place. Ribosomal RNA actually makes up these protein factories. And then transfer RNA are like trucks that deliver amino acids to the site of protein synthesis and brings them in the correct order so that they can be attached one to another to make a protein. All right, so protein synthesis occurs in two stages. There's transcription and translation. Transcription is this process where the gene is copied into a complementary sequence of RNA, and that typically makes a messenger RNA molecule. So as you can see here, during transcription, the DNA molecule is going to partially unzip just enough to expose a gene. So this area right here would be a gene. And an enzyme is necessary for this transcription to take place. This enzyme is called RNA polymerase. As the gene starts to unzip, RNA polymerase reads the gene sequence and starts to attach RNA nucleotides, complementary RNA nucleotides, in the correct order. And so one by one, these RNA nucleotides will attach together and create a long, single, straight chain of RNA nucleotides, and this is known as messenger RNA. So this process of copying the code of DNA into a messenger RNA molecule is known as transcription. Now, we've talked about the language of DNA, and once this copying is made into RNA, we can see that the four letters now in RNA are going to be A, U, C, and G. There will be no T's, remember. And these four letters can be combined into three-letter words Those three-letter words are known as codons. So for instance, AUG on messenger RNA would be a codon, and AAC would also be a codon. And these codons code for a specific amino acid. Now you may recall that there are about 20 different amino acids in living things. And so the order that these codons are arranged in will determine the order of how these 20 different amino acids are arranged. And so this is the code. This is what DNA ultimately is storing, the information that determines the order that these amino acids are laid down in, because this will determine the particular protein that's formed. There's a codon chart. One of them looks like this. You may have this one in your book, I believe. And this helps us to determine what amino acid goes with which codon. So, for instance, this codon, AUG, we can go to the codon chart, and we start here in the middle. The first letter is here, A, second letter, U, third letter, G, that codes for an amino acid called methionine. This codon, AAC, first letter A, second letter A, third letter C, AAC codes for asparagine. So you can see all the 20 different amino acids are represented by a number of different codons. Some amino acids are coded for by several different codons. There are actually 64 different possibilities here. One of those codons, the AUG, is always going to be the first, or what's known as the start codon. So AUG, methionine, is always going to be the first amino acid in a protein. And then there are some stop codons as well. There are 3 different stop codons, which tells the cell that it's time to stop the production of this particular protein. It's done. All right, once messenger RNA has been completed, the second stage of protein synthesis will take place, and that's called translation. In translation, what we're going to do is decode that message that's in messenger RNA into a particular protein. Now this takes place out in the cytoplasm on the ribosomes. So messenger RNA, once it's been transcribed in the nucleus, is going to leave the nucleus through these nuclear pores. And this long messenger RNA will come out into the cytoplasm, and it's going to find a ribosome and sit down and attach to a ribosome. And so here's our messenger RNA, here's our ribosome, and this is where and when translation will begin. So each tRNA molecule now, which is this molecule right here, it looks like a T. Each tRNA molecule is going to have an amino acid attached on one end, and on the opposite side it's going to have a 3-base anticodon, which is going to be complementary to the messenger RNA codon. So for instance, AUG is the codon, the anticodon is UAC. And the tRNA that has the anticodon UAC is just going to so happen to be carrying methionine, which is what AUG codes for. So each tRNA molecule is going to be specifically carrying the amino acid that's coded for by the codon on messenger RNA. So as this continues on, as tRNA continues to bring amino acids to the site one by one, as the amino acids are brought close together, they will bond to each other, forming a peptide bond. And this process will continue on down the length of messenger RNA one at a time, with amino acids combining with each other or to each other by peptide bonds. Eventually, forming this long chain of amino acids called a polypeptide or a protein. Eventually, the last codon will be a stop codon, which will cause all the tRNAs to detach and leave the protein fully assembled, ready to go do its job within the cell. All right, so that's the process of protein synthesis, transcription, and translation. We'll do lots of practice of transcription and translation so you can become very familiar with it. So if you have any questions, bring them to class or send them to me on my big campus. And looking forward to seeing you in class tomorrow. Have a super day.